Okay, there's the parasites we're going to talk about is these are all blood or tissue protozoa. I should qualify that not just blood, but some of them are tissue protozoa as well. We will talk about two forms of trypanosomiasis: the African trypanosomiasis, Trypanosoma brucei, that causes sleeping sickness, or and is prevalent in Central South Africa. Tens of millions of people are affected, and the major reservoir for this is domestic hoofed animals. So horses can be infected, and the deers are reservoir, and most importantly, cattle get infected, and there is a more economic burden than human suffering because of these organisms. Primarily in Central Africa. We'll talk about the distribution and other features later on. T. krusei, or the South American trypanosomiasis, and it is a causative agent for Chagas disease. If you are blood donors, most of you are, you will see on the form, have you ever had Chagas disease? And I sometimes chuckle because what percentage of population would really know what a Chagas disease is, if they have had it? But that's what is the common name for this infection, Trypanosoma krusei. And there are millions of people that are affected, and not only the people, but there are reservoirs for this: dogs, domestic dogs, and possums, raccoons, all sorts of wild and domestic animals can be infected, can be carriers for this. Leishmaniasis, primarily Mediterranean, the tropical form, but also India, Pakistan, they have had those Near East as well. Leishmania brasiliensis and others, they cause mucocutaneous leishmaniasis. They're more in South Africa. And Leishmania donovani is also in Asia. Okay, it's seen in Asia as well. And with our troops traveling abroad, more and more cases of these things are seen. You will see some of the epidemiology of these infections in later slides. Okay, the other organisms, Plasmodium, 4 different forms of Plasmodia, those 4 different species of those cause malaria, and it's one of the major causes of parasitic infection, blood infection. And some of it is often fatal if not diagnosed properly. Okay, so that's there's a lot of concern for this particular organism, both WHO and our military, as well as our NIH is spending a lot of money on research on this organism and development of vaccines. Toxoplasma gondii, again, it is causes toxoplasmosis, somewhat opportunistic. It can be dangerous in a normal individual as well, and we'll talk about that. What are the conditions one has to be careful? Some of you, I'm sure, would have heard about it, and we'll talk about later. Okay, Babesia microti is another organism that we'll talk about. It's Northern America, parts of America, North America, and Europe. It is found. Causes babesiosis, and we'll see the symptoms. So, coming to individual organism trypanosomiasis. African trypanosomiasis, also known as sleeping sickness. T. brucei rhodesiense is, and then the other one is T. brucei gambiense. The central to eastern part of Central Africa, and the Brucei gambiense more towards the west coast. But these organisms don't carry passports or need immigration or emigration visas. Therefore, they can be found across the borders as well of those countries that are in the center. And occasionally they are seen north and south as well. Our we are not at risk unless a person has been. And there have been cases, several cases in this country as well as Europe, where the person has been to those areas or lived those areas, particularly missionaries. So, and they get infected, or soldiers, military, missionaries, Peace Corps workers are obviously at risk if they are traveling those areas. Then American trypanosomiasis, as I said, Trypanosoma Chagas disease, and that's caused by Trypanosoma cruzi. So 3 names you've got to remember: Trypanosoma brucei, Gambiense, Rhodesiense, and then Trypanosoma cruzi in terms of trypanosomiasis. And also know Chagas disease and sleeping sickness is the sort of common names for those. The organism generally is sort of a spindle-shaped, undulating. It sort of moves by just undulating movement, almost like a fish. I have seen the organism because I had some in the lab, and I was infecting some mice to study their immune responses. Okay, so that's the general shape, and here is a sort of map of the distribution, geographic distribution. You can see Rhodesia is somewhere around here. Okay, that's where the Rhodesiense will be found, more towards the eastern part of the Central Africa, and then Gambia is somewhere over here, and it will be found on the western part, and you can see that there will be overlap in between. Okay, it has got 2 morphological forms depending on whether it is in the infective host, infective vector, or the human or mammalian host. This is trypamastigote form, and this is referred to as epimastigote. And the difference between the 2 is that the attachment of the flagellum is more sort of posterior region of the organism in the case of blood form, and in the case of the vector form, it is more towards the anterior of the nucleus, actually. Okay? The flagellum is attached to the body most of the way, leaving a little bit free flagellum for movement. Epimastigote, this is also referred to as crithidial form. So if you see crithidial, know that it is epimastigote. Trypamastigote is trypanosomal form, also referred to as trypanosomus form or trypamastigote. Okay, this is the vector. It's known as tsetse fly, TSE, tsetse fly. And it's found, of course, in those areas, the Central Africa. And the humans are, or the ungulate or hoofed animals. Ungulate, you'll see hoofed or divided hoof. Don't think all horses have divided hoof, but the hoofed animals really. So it can affect horses. Deer is depicted here, but horses and camels, if they're there, or cattle, they can all get infected. Okay, humans get infected via the bite of a tsetse fly while it's taking up meal. It has the organisms in the salivary duct, and it can infect the humans. Is it in the salivary duct, or is it in the esophagus, or the gut? No, it is in the salivary. Okay, so it can infect the humans. Okay, symptoms of African trypanosomiasis, depending on what the stage after the bite or after the infection they are. At the bite site, there is a skin reaction, and that is a non-pustular, itchy, and painful chancre, okay, meaning thickened, hardened lesion. Itchy, painful. However, the very important point is or an important point is that it leaves no scar. So if you have been bitten and for a few, while you're traveling in Africa, that part of the world, and it's a little painful or itchy, and you think it's like a mosquito bite or anything else, it has caused a painful chancre, but that disappears in a few days, and there's no scar, so you don't remember. If the physician thinks, well, or if you're taking history, they may not remember whether they have had it. Okay? So that's the after infection, there is a parasitemic stage when the organism is migrating to lymph nodes and blood circulation, lymphatics and blood circulation. And of course, when the lymph nodes are involved, there will be lymphadenopathy. And when the organism is in the circulation, there will be fever. And the reaction, the immune system will produce cytokines, of course, that will cause general unwellness, lassitude, sort of laziness, insomnia, lack of sleep, edema, also a feature of infection, an immune response, and a lymphadenopathy, painful swollen lymph nodes. Particularly important is one of the lymph nodes, at the back of the neck. I can't remember what the proper anatomical name is, but there's a back of we'll talk about that in a minute. Okay, CNS, it can migrate to CNS or heart. It has the predilection for going into the heart. And in both cases, of course, it will cause neurological problem that may be represented by personality changes, sort of reclusiveness, lack of interest in many things, the withdrawal from the social circle, lack of tremulous speech, mental retardation, sleepiness. Sleepiness, that's why it's called sort of uncontrolled desire to go to sleep. And cardiac failure can occur in this case. Now, sleeping sickness refers to as a sort of permanent sleep or prolonged sleep that may or may not occur depending which organism is infecting. And we'll sort of distinguish that in a minute. Okay, here's the lymph node swelling. You can see probably from the back as well, or certainly on your own monitors, on computer monitor, and is referred to as Winterbottom sign. And here's the sort of one picture of a coma that results from, of course, the infection of the CNS, and the person sort of goes into coma and does not wake up, does not come back, and that's why it's referred to as sleeping sickness. Now, T. brucei, pathology, inflammation, and inflammation due to the presence of organism, various cytokines being released, and also type 3 hypersensitivity because it produces antigens, it has antigens, and that they are shed off in the circulation in the body, and does produce an immune response, and so there is type 3 hypersensitivity. So presence of organism and type 3 hypersensitivity are the two causes of pathological changes. It can cause the presence of organism can also cause damage to the CNS. The antigenic change I'm going to describe a little bit further in a second. Okay, I think I have got some slides off it. Antibodies are not protective due to antigenic change. One would also see polyclonal activation of B cells. Polyclonal activation, you already know it, and that will result in immunoglobulin elevation, concentration of immunoglobulin. So elevated hyper IgM and even IgG levels are increased. Hypocomplementemia will be from immune complexes. Every time that you've got persistent immune complexes, you're going to have hypercomplementemia. And if they are not being cleared, which in this case I think they are cleared pretty effectively, there will be renal involvement as well and other blood vessels, characteristic of type 3 hypersensitivity. The organism also causes immunosuppression, and that is not totally understood what the mechanism is, but one of the mechanisms is induction of activated macrophages, activated RE cells, that in turn cause immunosuppression by producing immunosuppressive cytokines. So it does cause immunosuppression, and that will result in secondary infections as well. Okay, what I was referring to, antigenic change, I will describe. It has a very, very thick, diffuse, very tightly arranged glycoprotein that is called outer coat. Okay, and that is what the antigen is. One of the features of that antigen is it makes it complement resistant to complement-mediated lysis. Because remember, complement-mediated lysis occurs when there is a lipid membrane exposed. That's where the complement components, lytic components, are inserted. Having a thick coat is protected from complement-mediated lysis. That's one of the methods. But that thick coat has got antigenic features, and it induces an immune response, and as the immune response is increasing, the number of organisms will go down, but some of those organisms shed off that antigen and start producing another antigen. Same glycoprotein, but its structure is slightly different, and therefore it's not recognized by the previous immune response products, meaning antibody. So this is, I think, in animal studies, this cycle, but it happens in humans as well. In the laboratory, it has been shown that, okay, clone A is injected to infect the animal. There will be parasitemia, and it will peak. And during that period, of course, there is also production of antibody. Okay? Production of antibody, and this clone will be eliminated, but a few of this clone animals will change their coat and become a clone B. Okay? Same parasitemia and same elimination due to an immune response against this, and as it is going down, antigenic change will give rise to clone C, and D, and F, and G, and H, up to 100 have been reported. And it has the mechanism, it's out of scope of this particular class, but it has the molecular mechanism to bring in a new cassette of antigen producing gene and produce that gene. Okay? So that is one of the major problems, that why the immune system cannot get rid of it. And also, for all sorts of, despite all sorts of efforts to produce a vaccine for animals or humans, has been unsuccessful. Yes? Yes, it would, of course, it will. But at no stage you are really parasite-free. Okay? As one sort of clone is going down, you have got some reduction, as you can see, but the clones are, yes, and it will cause symptoms as this clone disappears, a new clone is emerging. And those will be reflected in relapsing circulatory system, when the organism is in circulation, meaning fever and chills and fevers. Okay? So yes, there will be some up and down of symptoms. Since this is produced from the mice, I don't remember the communications or papers that they did monitor the temperature, but I would expect that the temperature will change. Okay. Brucei diagnosis is based on travel, history, fly-by, and symptoms. and a blood smear and CSF, finding organism in blood and CSF. It's not always easy to find the blood in the blood or CSF, and if one does find in the blood, this is the blood form, trichomastoid form, trophozoite form, and that is what one would really see in the blood sample. If one does not find, and one really suspects that there is a, based on history and symptoms, one suspects that one can pass the larger amount of blood, larger amount meaning several cc's of blood, on a anionic resin column, where due to the charge of the organism, it will stick to those particles in that that column, and if you elute it, you may concentrate that blood. The other possibility is bioassay, and that can be done in the mice. Human organism does infect the mice, and as soon as the mice are infected, there will be enormous number of these in the blood circulation, and one can definitely, and organisms, depending on the organisms, as few as 100 or a few hundred organisms injected into mice can produce a fatal parasitemia, and the mice dies. Or EIA, immunoELISA technique, ELISA immunoassay technique for detecting antibodies in the person. Because if the person has been infected, they would have produced antibody, and that can be detected by immunofluorescence using the inactivated killed organisms in vitro on slides, or enzyme immunoassay using antigens, surface glycoprotein, VSG. By the way, often you will see abbreviation VSG for variable surface glycoprotein, that changing code. And I think I have indicated that in the handout, that abbreviation too. Okay, T. brucei. Okay, I told you that I was going to give you a contrast, and it is in the note, between rhodesiense and gambiense. Gambiense is more prolonged infection, prolonged disease, and therefore, while it can cause carditis, it can cause encephalitis, it is slow progressing, long-term disease, and more. That's why you see sleeping sickness in those infected with that organism. Whereas the rhodesiense is fast developing, progressing, rapidly progressing infection, and you see more cardiac failure. So before the CNS stage can be reached, they die of cardiac failure. Prevention would be, of course, there's no effective vaccine because it changes antigen codes. So the only precaution is really control of the vector. And those areas are often at the risk of losing the environmental greens and forests. They are sprayed, and they change in the ecology. They are sprayed with pesticides to control tsetse flies. If you are in that area, one tries best to avoid the fly bite, tsetse fly bite, by using repellents or netting. Suramin and pentamidine are the drugs of choice. Suramin for acute infection, or pentamidine for acute infection, and there's a drug, melarsoprol, which is an arsenic derivative that's more toxic, but it is used in more advanced disease. Acute with suramin, pentamidine, and then melarsoprol, which is much more toxic, but then one has to really put up with that toxicity for treatment of CNS infection. So that is the African trypanosomiasis, or sleeping sickness infection. And I have already emphasized the points that I would like you to remember. Okay? Morphology, those are also listed in the table at the back of the handout. Now, American trypanosomiasis is found in most of South America and even Mexico. And it depends on the presence of the vector. In this case, it's a trematode or kissing bug is the vector. So you can find the cases. And the cases have been reported. I will show you one case that was reported in Tennessee. And you will say, well, okay, it's only one case. But it depends on who sees, which one of you sees that particular case. So you should have, again, the knowledge. Not only that, but there is such a lot of influx of people from Southern American countries that they may bring in the infection. And the vector is present in the U.S. as well, and therefore we may get a good reservoir of that infection, and you will see that how it was. This is the one. This is interesting. The television education of lay population. A mother of an 18-year-old boy in rural Tennessee found a triatomine bug, and you'll see the picture of what the triatomine bug looks like, which she saved because it resembled a bug shown on a TV. This is Journal of Infectious Diseases report, so I'm not making it up. Okay? I do make up some cases sometimes, but I warn you of those. Okay? A bug shown on a TV program about insects that prey on mammals. The gut content of the bug were found by light microscopy and PCR to be infected with T. cruzi, Trypanosoma cruzi. Blood specimen obtained from the child in July and August were negative by microscopy and hemoculture, but positive by PCR and DNA hybridization. So the child had been infected. Suggesting a low level of parasitemia. Specimens obtained after benzodiazole treatment were negative. He did not develop anti-T. cruzi antibodies. 19 relatives and neighbors also were seronegative. 2 of 3 raccoons trapped in the vicinity had positive hemoculture. So it's not too far away that you might find a rare case. Now, obviously, the physician, whoever treated or saw that patient, knew enough to administer benzodiazole, which obviously cured the person. So, and then these last few months, MMWR reported this another case. In December 2005, a man aged 64 had a heart transplant in February. Now that is why it can be transplanted by blood transfusion, and that's why you ask the questions. Blood donors are asked the questions: Have you ever had Chagas disease? And that refers to Trypanosoma cruzi mediated infection. Okay. Hospital with anorexia, fever, and diarrhea of 2 weeks duration. The T. cruzi in blood and biopsy from heart were obtained, were seen. The patient had no identifiable risk for T. cruzi because the person had never traveled in those infested areas. Traveled to a country endemic for Chagas disease. He was seronegative for T. cruzi antibodies, but positive for T. cruzi DNA by PCR. So the modern methods, like that, can pick up the infection. After initiation of nifurtimox, that's the other treatment for Chagas disease, his parasitemia rapidly cleared. However, in April 2006, the patient died from complications attributed to acute rejection of transplanted organs. Remember, when we were talking about transplantation, you can have delayed rejection or rejection after infections. And actually, T. cruzi has been shown in some research to have antigens that cross-react with cardiac tissue. Okay? That I have not got in I may or may not have in my notes. And I'm not going to ask you about it, but you will, just so that you know that there is cross-reactivity. However, the source of infection tracked the organ donor who tested seropositive for T. cruzi. That was the accident victim, of course, and the antibodies by RIPA and tested borderline seropositive. Okay. Obviously, they had some blood sample from him. Otherwise, being a heart donor, he would not have we could not do serum immunofluorescence or any other test for antibody. He had been born in Mexico. He'd been born in the U.S., but had traveled to a T. cruzi endemic area in Mexico. So you don't know, unless you suspect, unless you are subconsciously aware of these infections and these critters, you can do harm to an individual, while trying to do good, meaning transplanting an organ. And it does not have to be heart transplant. It could be blood transfusion, or it could be just any other organ transplant. And there's no opportunity always to test those organs for the presence of the organism or presence of infection. Another case, January 2006, there were 4 or 5 cases reported, one after the other in MMWR. And this is another one. In January 2006, man age 73, received a heart transplant. The patient was readmitted in the hospital in February with fever, fatigue, and abdominal rash. A thin blood smear revealed T. cruzi, trypanosomatosis, and blood cultures were positive for T. cruzi. The patient had no identifiable risk factor for T. cruzi infection, meaning born in the infested areas or having traveled to the infested areas. He was seronegative, but PCR positive for T. cruzi, indicating recent infection. The patient's rash and parasitemia resolved after 10 days of nifurtimox treatment. Serial endomyocardial biopsies did not reveal trypanosome from the biopsy, of course. And he remained seronegative by immunofluorescence for T. cruzi. The patient died in 2006 from cardiac failure. Who knows whether that cardiac failure was for what reason? Okay. The source of infection was the organ donor who had been born in El Salvador and was residing in LA at the time of his death. Okay. Tested positive for T. cruzi. Or for his death, actually his own death. And they tested the other, of course they have got to track, since it's a cadaver organ, they have got other organs transplanted into other individuals, and you've got to follow those individuals. I can't remember, I would have remembered if any of them had become positive, but they will be followed for a long time. Morphologically, it's no different from Trypanosoma brucei. There is a trypamastogote form or epimastogote or critidial form. This one is in the blood circulation. This one can also be found in the blood circulation as well as in infecting bug. And this one is in tissue culture. Okay, and this is the one found in pseudocysts. We'll talk about those pseudocysts in a minute. And I think this is in the bug. It is in the bug, this one. Epimastigote or critidial form is the bud. Critidial form, critters. That's where you associate that it will be in the bug. Life cycle, there are a variety of reservoirs, but humans get infected when they get bitten by a general name is reduvid bug, or also referred to as kissing bug, because it tends to sort of be attracted towards the warm breath area on the face and bites there, and that's why it's referred to as kissing bugs. And the humans get infected, the organism from the bite site migrates to the lymph nodes, from there to the circulation in the muscles. It may form pseudocysts. It can infect the gut mucosal tissue, gut tissue, or even CNS, and it has a predilection for neuronal cells as well as the cardiac cells. And some of the carditis may be because of the presence of the organism, and some of it is also because of the cross-reactivity between antigens, trypanosomal antigens, and cardiac tissue. Reservoir, a variety of animals, include domestic as well as wild animals, from rodents all the way to possums and raccoons, armadillos, etc. Okay, raccoons and armadillos and possum. Okay, so it's it's very hard to get rid of the organism. And if you are interested in the disease, you'll pick up journals and you'll see a lot of news as well as cases from South America. It is a rural disease. It's not a urban. But the dogs have don't follow the socioeconomic taboo of traveling from poor area to rich areas, and occasionally there will be reduvid bugs in the affluent areas as well. These bugs have been found as far as Maryland, Tennessee. You saw that, the note there. Southwestern part of the United States also has those cone nose or kissing bugs. Okay, here's a typical example where there is sort of stone stacked and a thatched roof or straw walls and thatched roof, and there are no mosquito screens or on the windows. If they have window shutters, they are lucky, and that's the sort of living conditions. It can get into the house, and it tends to bite people at night. It can fly, but it's a more crawling, creeping bug, and it resides in those recesses in these walls, and at night it will come out and for a blood meal. Okay, transmission is through bite. I'm going to just go through this slide and then stop. Transmission those are the scientific name, tritoma. I would not distinguish between these different organisms, but tritoma, Rodnius, and Panistrongylus, if you just remember those words, you'll be doing very well. Tritoma is the general word, or kissing bug or cone nose bug are the other two terms used, and those are in your handout. And here's a picture of that sitting on a skin surface area. Cone nose because the sort of the head and anterior the stinging organ sort of make up like a cone. And I have already told you why it's called kissing bug. And here's a scenario, there's a bug sitting there and it's taking a blood meal. And while it is taking blood meal, it also has a very, very rude and bad-mannered habit of defecating. And if you think of it really, if you're asleep and something bites me, a bug bites you, or what's the first reaction? Huh? No. In your face, you just go like that and that, right? And that smears the fecal material that contains the organism into the open skin, broken skin, and organism enters the tissue and circulation through there. Okay? The symptoms, depending on... Oh, no. I promise you that I'll take a break after this. Okay, we'll continue tomorrow. No, tomorrow you do... Yeah, tomorrow. Tomorrow's Saturday.